Hello everyone and welcome to the session 5 of NPTEL course in pharmacognosy and phytochemistry. In this session, we are going to learn about a very unique pathway, and that is called as your shikimic acid pathway. Now this pathway is unique in a sense it is seen in plants and certain microorganisms, but it is absent in humans. But it is very essential for producing amino acids such as phenylalanine, tyrosine and tryptophan. These amino acids are often referred to as essential amino acids. The reason being because we miss out on this particular pathway in our bodies, we have to rely on dietary source for this amino acids. So let's learn what is shikimic acid pathway and what do we get out of it primarily and what secondary metabolites do we get out of them. So shikimic acid pathway is a secondary metabolite pathway which gives us primarily amino acids such as phenylalanine, tyrosine and tryptophan via a shikimic acid intermediate. So what happens is, if you see your biosynthetic pathway, you have your photosynthesis and then you have your Embden-Meyerhof pathway. Through Embden-Meyerhof pathway, we get what is called as phosphoenol pyruvic acid. Now this phosphoenol pyruvic acid is one of the starting material for your shikimic acid pathway. The second starting material comes from a shunt pathway called as hexose monophosphate. Through this you get a second compound which is a 4-carbon sugar phosphate, that is your erythrose 4-phosphate. So together your phosphoenol pyruvate and erythrose 4-phosphate are used to prepare the amino acids. Now this shikimic acid pathway via this amino acids gives us a number of building blocks. In the previous sessions we have learnt about the different building blocks. So what building blocks do we derive from the shikimic acid pathway? The first building block that we can derive from the shikimic acid pathway is your phenylpropanoids. The second one what we can derive is what you call it as phenyl ethyl amine unit, and the third amino acid, that is your tryptophan, contributes to what is called as indole C2N building block, that is the tryptophan or often referred to as the tryptamine building block. So we get C6C3, we get C6C2N and we get indole C2N as building blocks for our secondary metabolites from this essential amino acids. Now if you go to see these building blocks are there in numerous organisms. So shikimic acid pathway you can see in bacteria, you can see in fungi, you can definitely see it in plants, algae and some protozoans. Now this helps not only in building amino acids for them, this helps also provide vitamins and some defensive molecules. Now coming to secondary metabolites, in terms of secondary metabolites, your shikimic acid pathway gives you compounds such as tannins, cinnamic acid derivatives, your lignans, lignins, the glowing molecules, that is your coumarins, the antioxidant molecules, that is your flavonoids and anthocyanins, and certain alkaloidal compounds are also derived from your shikimic acid pathway. Now to start with, your shikimic acid pathway happens via two starting materials, phosphoenol pyruvic acid, as you can see here, that is your phosphoenol pyruvate, and the second molecule is a 4-carbon member sugar, that is your erythrose 4-phosphate. So we add a C3 and a C4 to get a 7-carbon unit. This 7-carbon unit is called as deoxy-D-arabinoheptulosinic acid 7-phosphate. So what happens, you can focus here. So if you see this carboxylic acid, this is the carboxylic acid, the phosphate is removed to get a ketone, and here you can see this carbon. So here you have this 1, 2, 3, I am just labeling it here 1, 2, 3. Now this joins with the 4 This is your 4. What happens here in the 4 is your aldehyde is now converted into an alcohol. So here you have your 4, 5, 6 and there you have your 7. That is a 7 carbon unit and that is why you call it as heptulosonic acid derivative. Now it is a heptulosonic acid 7 phosphate. So the terminal carbon, the 7th carbon carries a phosphate. Now this is involved in a condensation reaction and this condensation reaction in presence of enzyme again a synthase, that is a dehydroquinate synthase cyclizes to give you what is called as dehydroquinic acid. You can see here the cyclization has happened here and during this process this particular ketone has got converted into an alcohol. So you can see this alcohol moiety that is contributed by this moiety and you get what is called as dehydroquinic acid. Now from dehydroquinic acid you can get your quinic acid derivative. Now dehydroquinic acid is an important intermediate. If you see compounds such as your tannins, especially your gallic acid derivatives, if you go to see gallic acid has a very simple structure. So there is a good resemblance between dehydroquinic acid and gallic acid. So what you need to do is eliminate this, probably reduce this and aromatize this. So this is a multi-step process and eventually from your quinic acid you get gallic acid derivatives. So you have quinic acid or dehydroquinic acid that gives rise to some hydrolyzable tannins. From gallic acid you can get your ellagic acid and other compounds as well. In some cases what happens is this hydroxy undergoes elimination along with this hydrogen resulting in a double bond. So this happens in the presence of enzyme called as dehydroquinate dehydratase. Now when this happens you get a double bond here and the compound is your 3-dehydroshikimic acid derivative. Now this shikimic acid derivative, especially your 3-dehydroshikimic acid derivative, is then kind of reduced with the help of enzyme called as shikimate dehydrogenase and you get your main compound from which the biosynthesis is labeled. The compound is your shikimic acid. So this shikimic acid is kind of marker for plants or for organisms in which this biosynthetic pathway exists. Now this shikimic acid pathway doesn't stop here. It goes ahead to produce numerous amino acids that we discuss. So let's see them. So this shikimic acid undergoes what is called as a phosphorylation reaction at the 3rd position. So this is the first, second and we call it as 3rd position. So it undergoes phosphorylation at 3rd position to give you what is called as shikimic acid 3-phosphate in presence of an enzyme shikimate kinase. Kinases are generally the enzymes which are involved in phosphorylation. So now once you get the shikimic acid phosphate, what happens next is an interesting phenomenon. After phosphorylation, one more molecule, so additional molecule of an phosphoenol pyruvate, if you remember this was your C3. So one more C3 gets added. This happens in presence of enzyme that is enoyl pyruvyl shikimic 3-phosphate synthase. So this gets added and you get the compound which is your enoyl pyruvyl shikimic 3-phosphate. You can see that your pyruvic acid gets added here precisely and these are your 3 carbons. Now in order to undergo further processes, what happens is a dephosphorylation reaction. So this loses on its phosphate and as it loses on its phosphate you can see the changes in the unsaturation happening. So this happens in presence of chorismate synthase and you get what is called as chorismic acid. So from shikimic acid your next derivative is your chorismic acid. So from chorusmic acid we get an interesting set of amino acid called as tryptophan. So this chorusmic acid sometimes further undergoes amination reaction. So if you see on a phenyl ring, when there is a COH and NH2, we call this compound as an anthranilic acid derivative. So this anthranilic acid derivative is formed after elimination of these two groups. So this anthranilates are compounds which come from chorusmic acid, and in some cases what happens is amination happens here also, and this amination gives rise to molecules such as para-aminobenzoic acid. PABA are very important metabolites for microorganisms and also involved in vitamin synthesis. So you get your anthranilic acid, you get your PABA, especially from chorusmic acid. Now what happens once you get anthranilic acid, this anthranilic acid will add a sugar, especially your phosphoribosyl pyrophosphate sugar, and later on through a sugar opening reaction form an amino acid tryptophan. So it does a sugar opening, there is a transamination, and it is a multi-step reaction, and eventually what you get is an amino acid tryptophan. So from your chorusmic acid we have our first building block, and if you remember, this building block is what gives us our indole C2, that is C1, C2 and N after decarboxylation. So you get your tryptophan, from tryptophan you get numerous alkaloids being there. Now other thing what can happen from chorusmic acid is sometimes the phenyl pyruvic acid which has attached here shifts on to move to this position. Now this happens in the presence of an enzyme called as mutase. So chorusmic mutase orchestrates this event, and what you get is your compound moving here. So the point of attachment is this. So now if you carefully assess, this carbon is what gets attached here. Your oxygen gets converted into a ketone moiety. So you have this carbon, you have this oxygen which gets converted into a ketone moiety, and you have your carboxylate. So here if you see carefully, you have your C6, C3 almost in line, but one carbon is extra. Now this carbon is eliminated along with water through an enzyme called as prophenic acid dehydratase. So this elimination happens and you get what is called as hydroxyphenyl pyruvic acid. Now depending, during the elimination this hydroxy may stay or this hydroxy may get eliminated. So if the hydroxy stays, you get what is called as hydroxyphenyl pyruvic acid. If the hydroxy gets eliminated, you just have what is called as phenyl pyruvic acid. Now why this is important is, now the next reaction to this is a transamination reaction. During the transamination reaction, this ketone moiety gets aminated. So if this hydroxyl is intact, you get amino acid tyrosine. But if this hydroxyl is missing, that is from phenyl pyruvic acid, you will get your phenylalanine, which is nothing but tyrosine missing a para-hydroxy group. So from this particular way, that is from chorusmate, you could get your tryptophan, you could get your phenylalanine, as well as you can get your tyrosine. So all these amino acids are involved in preparation of numerous primary as well as secondary metabolites. So let's see what are this. So if you see your phenylalanine, phenylalanine is something which helps to contribute along with tyrosine in preparation of plant pigments such as flavonoids, isoflavones, your anthocyanins, proanthocyanidins, which later on get converted into condensed tannins. Not only that, it helps you in preparation of lot of defense compounds. This includes your cutin, suberin, lignin, lignans, as well as your cinnamic acid derivatives. Now talking about tyrosine, tyrosine is also one of the important amino acid which is essential. The reason being, it is involved in biosynthesis of numerous pigments as well as neurotransmitters. Neurotransmitters are very vital for conduction of neuronal signals. So it is involved in synthesis of L-dopamine, dopamine, epinephrine, norepinephrine, phenylethylamine as well as tyramine. In terms of pigments, it is involved in formation of melanin-like pigments, which are your eumelanin and pheomelanin, which are also thought to be your melanin precursors. Now coming to tryptophan, tryptophan is involved in synthesis of vitamins and numerous cofactors such as niacin, NAD, NADP. It is involved in plant hormones such as auxins, indole, acetic acid derivatives, tryptamine, and also in presence or also involved in synthesis of alkaloids. Your amino acid tryptophan is also involved in preparation of neurotransmitters such as serotonin, melatonin and tryptamine. Now coming to secondary metabolites, let's see how it is processed further. Now this amino acids are your primary metabolites. They need to be prepared in order to undergo or to divert from their primary metabolism and to go into secondary metabolism. So this happens in an enzyme called as phenylalanine ammonia lyase if it is a phenylalanine, or tyrosine ammonia lyase if it is a tyrosine. So lyase is an enzyme which cuts off. So we need our amino acids to be converted into something like a phenylpropanoid derivatives, wherein if we don't want amino acids, in that case the ammonia lyase will take care of it. Now that the amino groups have been reduced, you get what is called as tranexamic acid, or in this case hydroxytranexamic acid derivatives, and later on they will get converted into coumaric acid derivatives. Okay. So hydroxytranexamic acid derivative is called as coumaric acid derivative. Now you can derive that directly from tyrosine via tyrosine ammonia lyase, or via phenylalanine, where it first gets converted into cinnamic acid and later on gets hydroxylated in presence of enzyme called as tranexamic acid 4-hydroxylase to become coumaric acid. Now this coumaric acid, once it is ready, gets added to an enzyme called as coenzyme A. This happens in presence of an enzyme that is 4-coumaryl-CoA ligase. Ligase is something with joints or sticks. So once this ligase acts in, you have your para-coumaryl coenzyme A. Now this para-coumaryl coenzyme A is an important precursor. Now if you see that in terms of nuclease, you have your C6, which is your phenyl ring. You have your 1, 2, 3, C3. So all your C6, C3 derivatives can be borrowed from this coumaryl-CoA via different mechanisms or different enzymatic pathways. The important compounds that we get from this are your lignans, which are nothing but C6, C3 into 2, lignins, which are C6, C3 into N. That is what happens here is your lignins are nothing but polymers of C6, C3. So what you can do in this case is your polymers C6, C3 and just multiply them n times, you will get your lignins. You can get your compounds such as coumarins which show fluorescence, and you can get cinnamic acid derivatives which show you a very good defensive action because of their acidic nature. So let's see them. Cinnamic acid derivatives, so once you have your coumaryl-CoA, your coumaryl-CoA gets oxidized to coumaric acid. So this is your coumaric acid. So this coumaric acid further undergoes hydroxylation in presence of an enzyme called as coumarate 3-hydroxylase. Now this coumarate 3-hydroxylase will add a hydroxy group out here and give you compound called as caffeic acid. Now this caffeic acid will undergo methylation. You remember your building blocks? Now this can be done with the help of an enzyme methyltransferase. Now this methyltransferase can be depending upon the substrate. Whenever it is a caffeic acid, it will be caffeic acid O methyltransferase. Okay. So COMT is caffeic acid O-methyl transferase. Now once this methylation happens, you get a compound called as ferulic acid, and then you have hydroxyferulic acid. Now this is again done by ferulic acid hydroxylase. Now what has happened here is, this is your position 1, 2, 3. This is 4 and 5, and that is the reason it is ferulic acid 5 or ferulate 5 hydroxylase. Now once you do that, you can even methylate it. Now in this case you can have your hydroxyferulic acid O-methyl transferase, and you will have your cinnamic acid being present. So cinnamic acid, ferulic acid, caffeic acid, coumaric acid are all cinnamic acid derivatives which play a very vital role in defense because of their acidic nature. Now what happens in plants is, these acids can also get converted into alcohols. So you have your coumaric alcohol, wherein this will get converted into CH2OH. For all of them, you will have your ferulic alcohol, you will have your synapyl alcohol. So these compounds will also get converted or reduced to their corresponding alcohols in plants, and these alcohols can give rise to new set of metabolites. So cinnamic acid derivatives are produced from your coumaryl CoA. Then the next set of compounds, like I said, because they are acids and they can be reduced, here you have 3 compounds which we take from them: coumaryl alcohol, coniferyl alcohol and synapyl alcohol. Your coniferyl alcohol came in from your ferulic acid. So if you see this carefully, your ferulyl alcohol or your coniferyl alcohol is nothing but the same COH being converted into CH2OH. So coumaryl alcohol, coniferyl alcohol and synapyl alcohol undergo condensation, and this condensation, if it is a dimerization, the compounds thus formed are called as lignans. So lignans are the compounds formed by dimerization of most of the what are called as cinnamic acid derivatives, later on reduced to their alcohols. So this alcohols you can see very clearly here. This is a dimer of coniferyl alcohol. So what happens is, your double bond which was there is now involved in formation of this bond, and 2 molecules of coniferyl alcohol gives you a lignan derivative called as secoisolaricyresinol. Similarly, sometimes what can happen is, these compounds or these substituents may cyclize in themselves and give rise to compounds such as your martiaresinol. And sometimes a rearrangement plus addition helps in, but you can still see that the phenyl propanoid nucleus is maintained. So this is your phenyl, and you can see 1, 2, 3. This is your phenyl, and here you can see a 1, 2, 3. So they are still phenyl propanoids, and this little adduct comes because you can see your methoxy that has been involved in kind of cyclization in that. This is compound sesamin which is present in your sesame seeds. Lignans are very good for health and thought to be phytoestrogenic in their action. The next set of compounds are lignins. So if you take this alcohols, they are also referred to as monolignals. Dimers will give you lignans, but the polymers will give you lignins. Lignins are involved in strengthening of stem, and what happens is, once you have your phenyl propanoid compounds, especially in form of alcohols, the monolignals are transported into cell wall, and once they are transported into cell wall, they are kind of polymerized in themselves through radical coupling. Here you have unsaturation, so it is very easy to initiate the coupling, and there are enzymes such as lactases and peroxidases which further converts them into free radicals and help them in the polymerization reaction. As a result, you have a big kind of C6C3 or monolignol addition products, and these are called as lignins. So as you can see, as discussed from the previous session, you have a lot of your C6C3 being added to give you a huge polymer, and that polymer is your lignin. The next set of compounds which are derived from your phenylpropanoids are your coumarins. Now coumarins are cyclic compounds, they involve lactonization. So what happens here is from phenylalanine and tyrosine you get a compound called as coumaryl CoA that we have discussed. Now this coumaryl CoA further undergoes hydroxylation, and this hydroxylation is done in presence of an enzyme called as hydroxylase, and further it undergoes lactonization, that is it will undergo cyclization. Now when it undergoes cyclization, from here you will see this will join and you will get a coumarin, that is a lactone like structure, and this is catalyzed by an enzyme called as coumarin synthase. So if you can carefully see here, this is your tyrosine. So you can convert your tyrosine into coumaryl CoA, then it is hydroxycoumaryl CoA, and then when you cyclize it from your coumaryl CoA you will get your umbelliferone. In some cases your coumarins can be substituted. So instead of having your coumaric acid, if you have seen previously in terms of monomers, you had your coniferyl alcohol. Suppose coniferyl alcohol is the compound to be cyclized. In that case, what will happen is you have a hydroxy, you have your methoxy, and then involve this in cyclization. So this is what you get from coumaryl alcohol is scopolamine. So cyclization of different type of phenylpropanoidal alcohols gives you different coumarin derivatives. Not only that, this coumarin derivatives may add with other small metabolites to form furanocoumarins, pyranocoumarins and so on. The next set of compounds which are obtained from phenylpropanoid derivatives are slightly different, and in this case it is not C6C3 but it is C6C2N. Now C6C2N is referred to as your phenylethylamine unit. Now this happens when decarboxylation is carried out. So rather than we losing your amine when we had your phenylalanine ammonia lyase, we removed out the amine. In this case, we will not remove the amine, but we will remove the carboxylic acid. So the decarboxylation reaction converts them into compounds such as phenyl. This is 1,2, so we call it as phenylethylamine derivatives. And phenylethylamine derivatives are present in numerous alkaloids. To give you a few examples, as we have previously discussed, I am keeping it same for your reference. So your mescaline is a good example of a phenylethylamine, and so is your analgesic compound which is present in opium, that is your morphine. So from morphine also you will have your 1, you will have your 2 and you will have your amine, that is the next group. Not only that, if you see your amino acid tryptophan, that contributes to your indole C2N. Now this indole C2N we visualized last time. This is your indole moiety and this is your 1, this is your second and this is the N. So this is your indole C2N. Now what happens in the plant is your indole C2N condenses with different set of compounds. This compounds can be something like your secologanin in case of reserpine. So a series of enzymatic transformation follows. So you can see a secologanin, you can see a tannin, that is something like a trimethoxygallic acid derivative, and later on all of them condense together to give you a reserpine. Now reserpine is a hypotensive agent which is found in your Rauwolfia species. So next set of compounds which we obtain are not clearly or pure phenylpropanoid derivatives, but they are adducts. So half of these compounds have been taken from your Shikimic acid pathway and half of these compounds have been derived from other pathways, and these compounds involve your condensed tannins and flavonoids. So what happens in this case is your coumaryl CoA condenses with 3 units of malonyl. So from your previous building blocks, if you recollect, malonyl is a reactive form of acetyl CoA. So when acetyl CoA adds up, you get a C2 building block. So when you have your 3 malonyl CoA, you have 3 into C2, which is nothing but your C6. So you get what is called as a phenyl ring. But if you remember, acetyl CoA has a aldehydic group. So you have an alternate all to it. So this is nothing but this part of it is contributed by your coumaryl CoA, and this what happens is, to this coumaryl CoA you will have your first, that is from here, you will have your second and you will have your third acetyl group being added in form of malonyl CoA adducts. This condensed together initially in form of a chalcone synthase to give you chalcones, which is an open ring structure, but later on gets converted into a closed ring structure in presence of enzyme which is called as chalcone flavone isomerase. Now this flavone isomerase can further convert this flavonoids into different set of flavone derivatives, either by adding hydroxyl groups or by adding unsaturation. So series of enzymatic process follows and you will get your numerous flavonoid compounds. Now in some cases a hydroxy group can also be introduced by a simple enzyme hydroxylase, that is your flavanone 3-hydroxylase. In that case you will get your 3-hydroxy substituted flavonoid derivatives. 3-hydroxy substituted flavonoid derivatives are very potent antioxidant agents, and if you kind of unsaturate it, you get compounds such as your quercetin, you get compounds which are involved in formation of flavonoid glycosides. So extremely polar compounds can be obtained when you hydroxylate your flavone, especially at the third position. So from your from your shikimic acid pathway, you get numerous secondary metabolites, and some of this are not pure, but some of this can be adducts. Here are few references if you wish to dwell more into this biosynthetic pathways. And thank you everyone. See you at the next week.